Testing. Hello, everybody. So, last week, we talked about the resting membrane potential and then how that then leads to the creation of the action potential. So, which ion most contributes to the resting membrane potential? Which ion most contributes to the resting membrane potential? Potassium, right. And so, what ion is going to be the main trigger at the beginning of the action potential? Sodium, right. And what kind of channels are going to open up to allow sodium to come in? Is it a resting channel? Is it a gated channel? And what kind of gated channel? Yep, it's gated. And what kind of gated channel? Huh? No, remember there were two types. There was voltage gated and chemically gated. Voltage gated, right. Because we're going to have to reach that threshold of excitation. When we have a voltage change of about 15 millivolts, that's the trigger that's needed to open up those voltage gated sodium channels. It's going to allow sodium to rush in. So sodium, what kind of charge does sodium have to it? Positive, right. So that's going to allow positive sodium to rush in, and that's going to depolarize the cell, taking it from its resting membrane potential, which is normally negative or positive? Negative, and it's going to make it positive. All that positive sodium is going to run in, make the cell go positive. And then what's going to open up after the voltage-gated sodium channels? What kind of channels are going to open up? Potassium channels, right. And so then potassium, positive charged potassium is going to leave the cell, and it's going to allow it to repolarize. And so that's your action potential. And then it's going to reset, and it'll all go again. And so that action potential is going to then propagate down the axon, and from and it's either going to go down a myelinated or an unmyelinated axon. Which one's faster, myelinated or unmyelinated? Myelinated is going to be a faster one to go down. Because when it goes down the unmyelinated, those channels are going to constantly be opening up sodium, potassium, sodium, potassium, sodium, potassium. And so that takes time for all that to happen. As in an unmyelinated, the opening, closing of ion channels is only going to happen in what areas along the axon? The nodes of Ranvier, right. And so that's, and then in the myelinated sections, then it's just going to be carried on through passive conductance. And so therefore, they'll move much faster. But if you didn't have the nodes of Ranvier there to regenerate it, then the signal would get lost. So the nodes play an important role in boosting the signal along the way. But the myelin allows it to travel faster. The other advantage to myelin is it allows for a smaller axon. You see, when you have an unmyelinated axon, the way that you can conduct information faster is by having a larger tube. Has to do with cable properties and physics. So if you don't have the insulation on there, you can have a larger diameter axon for information to move quicker. But in mammalian species, we have incredibly complicated nervous systems, especially in comparison to, say, invertebrates. Invertebrates who would have an unmyelinated system don't have as many neurons, and therefore they can afford for each neuron to be a little bit bigger, or for the axons to be a little bit bigger. But since we have many, many more times neurons, we got to fit them still into a limited space. So we need the very thin axons, so we can fit these billions and billions of neurons. We can fit their axons still in our bodies. So we can't afford to have large diameter axons. So myelin allows us to be more efficient in both speed and in space, that we can fit more neurons in a tighter amount of space by having myelin, because they can still be fast, but yet be smaller diameters. So myelin gives us several advantages. Okay, so we have the action potential. It travels down to the end of the axon to terminal arborizations, and at the end of the terminal arborizations, we have the terminal boutons. And so somehow we have to get that information from one neuron to the next neuron, or to a muscle, or to a gland. I'm going to talk about it in terms of communicating from one neuron to another, but realize it's all really the same process, whether we're talking to a muscle or to a gland. Now, initially, people thought the action potential just simply traveled from one neuron to the next. And you could imagine it's just somehow the positive charged sodium ions could just keep traveling, go from one cell to the next cell. Now, that is possible. There is something called a gap junction, which the two neurons would actually be physically touching each other, and there are membrane pores that would transverse both membrane and would allow, say, those positive charged ions just move from one to the next. This is a small minority, a very, very small minority of axons. It exists, but that is not by no means should that be thought of as a typical way that neurons communicate with each other. That's a very rare exceptional case. But there was a time when people thought that was just how it worked. Mainly because it was more an intuitive thing. They didn't have necessarily experimental evidence. They just knew, hey, these things seem to work on electrochemical signal. How could they possibly talk to each other? Oh, somehow this signal must be able to jump from one another. Just like we take a plug and we stick it in, you take your internet plug and you stick it in the back of the router, to get the internet in your house. And so somehow the electricity, the electrical signals just go from your router to your plug and then go from the plug to your computer, and so not that they were thinking about internet in the early 1900s, but, it just intuitively made sense. But then we started realizing that there was a chemical process in there. So the typical way that one neuron is going to communicate with the next neuron is going to be by using neurochemicals or neurotransmitters. So somehow that action potential has to come down and trigger a release of neurotransmitters. And so we're going to have a new ion I'm going to throw at you, calcium. So calcium, it's actually 2+, not just 1+. It has 2 positive charges to it. Not that it's that important for this, but now, anytime that you talk about calcium, and calcium's going to enter the cell, something's going to happen. Calcium seems to trigger things. There's different times when calcium will come in. We'll talk about it at other times. But pretty much anytime you're going to deal with the calcium ion, you should be thinking, okay, something's about to be triggered. Now, I'm going to give you the really short and dirty version of this. You can take a biology class and you can learn all the intricacies of this. But basically, an action potential, when it travels to the end of the terminal bouton, is going to trigger a new type of voltage-gated channel. It's going to trigger a calcium voltage-gated channel. And when these calcium channels open, calcium is going to come into the cell, and it is going to trigger what we call synaptic vesicles. Now, synaptic vesicles are made of the same thing, the same lipid bilayer that the cellular membrane is made out of. Except they're just a small little spear of that. And inside that spear is going to be a neurochemical. It could be acetylcholine, it could be dopamine, it could be GABA, it could be serotonin, it could be what have you. And so these synaptic vesicles, these little spears made out of the same lipid bilayer, is going to be triggered by the influx of calcium to migrate to the edge of the neuron. And so what's going to happen is basically they're going to dock. This synaptic vesicle is going to dock with the membrane, and it's going to become part of the membrane. So it's going to fuse with the cellular wall, and as it does that, it's then going to release whatever chemical is contained within it into the synaptic cleft. So it fuses with, it becomes a part of, and then as it does that, all the neurochemical in there is then going to be expelled out into this synaptic cleft between the presynapse. Let's use a little terminology here. We'll call this first neuron here. Oops, that's not what I wanted to hit. We'll call this first neuron here the presynapse. And we'll call the second neuron here the postsynapse. So we're going to have the neurochemical released from the presynapse, and then it's going to migrate across the synaptic cleft to bind with receptors that are located on the postsynapse. So this is how we're going to get the information from neuron 1 to neuron 2. The action potential is going to travel down the axon to the very end, where the action potential is going to trigger voltage-gated calcium channels which is going to allow calcium to come in, is going to trigger synaptic vesicles to then migrate to the edge of the neuron, fuse with the cellular membrane, and then release their neurotransmitters into the synaptic cleft. And then those chemicals will then migrate across the synaptic cleft to bind to receptors on the postsynapse. Here's a photomicrograph showing that. You can see this is the inside of a axon. Here's the synaptic cleft. Here's the synaptic vesicles, and here you can see where they're fusing with the cellular membrane. And so, too small to be able to see would be the chemical contained within it being expelled in here into the synaptic cleft. So on the postsynapse side, we have these receptors. Call them postsynaptic receptors. So we have molecules of neurotransmitter that bind to the receptors located on the postsynaptic membrane. And these receptors are going to open postsynaptic ion channels. Now, without me saying, if you had to guess, what kind of ion channels would these be? Would they be open, resting or gated? It probably makes sense that they're gated, right? Or because if they're resting, why does anything need to trigger them? Because they're just always open. So they're gated. And would they be voltage gated or chemically gated? Chemically gated, because we're going to have neurochemicals coming to trigger them to open. So now we're dealing with our first chemically gated ion channels. So neurotransmitters are going to bind to receptors, activate ion channels, then ions are going to throw through the membrane, producing either a depolarization or a hyperpolarization. So this creates what we call a postsynaptic potential. Remember, we've talked about the resting membrane potential. We've talked about an action potential. Now we're going to have a postsynaptic potential. And they come in two varieties. They can be depolarizing or hyperpolarizing. Remember, depolarizing means that we make the cell more positive, and hyperpolarizing means that we're going to make the cell more negative. Okay, let's talk about the receptors a little bit. There are two different ways that a receptor can activate an ion channel. It can do so directly or it can do so indirectly. We call the direct method an ionotropic receptor, and we call the indirect method a metapotropic receptor. Though sometimes you hear people use the term second messenger. And that's because that's what it uses. Basically, in the direct method, the ionotropic, the neurochemical is going to be that's going to be the messenger. That's what's going to trigger the ion channel to open. But in the indirect fashion, in the metapotropic fashion, the neurotransmitter is actually going to trigger the creation of a second messenger. And that second messenger is going to be the chemical that actually triggers the opening of the ion channel. So by far the simplest one to explain is the ionotropic receptor. So here, the receptor and the ion channel are part of the same protein structure. So what happens is that the neurotransmitter comes and binds to the receptor. And that's going to cause a change in the energy of the protein. Basically, remember last time I described that proteins are just these long chains of amino acids that fold up into a three-dimensional shape. When they do that, the direction that they fold up is where they're most happy. It's the place that doesn't cause any strain. It's the place where the protein's going to be relaxed. But if you go and mess with it, like add a new chemical in, that's going to cause some strain. That's going to cause the protein to no longer be in its optimal configuration. And so when you add a new chemical in, that's going to cause it's going to cause a conformational shift in the protein. In this case, what that's going to do is it's going to cause it to open up its ion channel. And when it opens up its ion channel, the ions are free to flow through. Now, there's an advantage to this, is that it's very quick. As soon as the neurotransmitter binds, it's going to open up. So if we need to get a signal through quickly, this is a good way to go. The downside to this is that this neurotransmitter is not going to hang there forever. It's going to come in, do its job, it's going to leave. So it's a very short-lasting effect. So it's quick. As soon as it binds, it opens up, the ions are flowing through, but it's very short-lasting. Now let's compare this to the metabotropic receptors. Without me even explaining it, you can already tell it's more complicated. Okay. There's not just one type of metabotropic receptors. There's actually a bunch of different types. This is just 2 examples, but there's a bunch of different ways that this could actually happen. The basic gist, though, is that the neurotransmitter itself is not what actually causes the ion channel to open. The neurotransmitter just simply starts a chain reaction that will eventually lead to the receptor from opening up, or to open up. So in this is probably about the simplest example of this, that the neurotransmitter binds to a receptor. The receptor then triggers a G protein, and this G protein will have a conformational change which will cause the separation of an alpha subunit, and that alpha subunit is what will trigger the ion channel to open up. So unlike the ionotropic, where it's the neurotransmitter that caused this to open up, here it's something different. It's a second molecule. It's a second messenger that's causing this ion channel to open up. Probably a little more typical would be that a neurotransmitter binds to a receptor. That receptor then communicates with a G protein. You then have an alpha subunit, which is going to dissociate, then going to trigger an enzyme. And that enzyme is then going to allow the creation of a second messenger. And that second messenger then is what's going to open up the ion channel. So one disadvantage to this system, as you can probably imagine, is if the ionotropic was a quick way to open up an ion channel, this has to be slower. Because if the ionotropic neurotransmitter binds, channel opens up, ions flow. But here, neurotransmitter binds the receptor signals G protein signals enzyme. Enzyme starts creating new chemicals. These new chemicals get created. They come over here, bind to channel, now channel's opening. So it's a slower mechanism to open up an ion channel. But the advantage to this is that one neurotransmitter is actually going to produce a lot of second messenger. And so all these second messengers, one's going to bind there, then another one, then another one, and another. So it's going to keep the channel open longer. So where the ionotropic was fast but short-lasting, the metabotropic is going to be slow but long-lasting. The other advantage to the second messenger system is this second messenger doesn't have to just communicate with this ion channel here. There's a lot of those being made. And so they can migrate to other parts of the cell and send signals to it. It can migrate to the nucleus. It can create new proteins or trigger the creation of new proteins. Because let's say, for instance, that someone is taking a drug. Let's say something like a drug of abuse. And so those drugs are interacting with these receptor systems here. They're acting as if they are the neurotransmitter. Or they're making your body release more neurotransmitter than it normally would. So that means this system is getting hit more often than it normally would. Okay, so I take cocaine, and my body's being flooded with dopamine. So my dopamine receptors are getting hit way more than they normally would, which means I'm creating a lot more second messenger than I normally would. And that means a lot more second messengers reaching the cell nucleus than it normally would. And my cell's going, what the heck am I getting all these second messengers for? Why am I being communicated so much? We need to do something about this. We need to remove receptors out. And so that triggers mechanisms that's going to go and then remove, because your body likes things to remain the same. And if you start overstimulating a receptor, your body's going to respond to that by removing away receptors. Hey, something's wrong. There's too much dopamine. I'm getting signaled too much. How do I respond to that? Well, just remove receptors. And if my receptors aren't there, I can't be triggered. And now we've given one of the mechanisms for tolerance. Because now the next time I take, cocaine's not a good example. You don't really gain tolerance to cocaine. Amphetamines. Now I'm going to need a larger, same dopamine release. So now I'm going to need a larger and larger dose of amphetamine to get the same rush from that. Because now I have fewer and fewer receptors. And then my body's going to take away more receptors. I'm going to need a larger dose and so on. So that's one of the mechanisms for how drug tolerance works. So, having this, and it works the opposite way too. Maybe I'm not getting signaled enough. And so the fact that the lack of second messenger working there might be a trigger that, hey, maybe I need to put more receptors out there. So there's advantages to this metabotropic system. Even though it's a more complicated system, it can do other things that the ionotropic system can't do. Okay. Now, once we open up these postsynaptic, or once we trigger these postsynaptic receptors that then open up these postsynaptic ion channels, we can have different ions flow in and out. It's the same ones we've been dealing with before. We're still dealing with sodium, still potassium, still chloride. So we haven't changed that. And as I've already mentioned, you're either going to have excitatory postsynaptic potentials. An excitatory postsynaptic potential is one that depolarizes the neuron. And we can have inhibitory postsynaptic potentials. And those are going to be ones that make the cell more negative. And if you think about it, that makes sense. Hey, whole new box of chalk. So remember, we have our resting membrane potential. And remember, in order to create the action potential, we had to depolarize the cell until we hit that threshold of excitation that would cause the action potential to happen. And this is the part that I said, just take my word on it. Well, now you might be able to figure it out. These are the excitatory postsynaptic potentials. This is the buildup of that. And if the threshold of excitation is what triggers the action potential, then depolarizing the cell, making the cell more positive, means that you're going to be more likely to fire an action potential. And hyperpolarizing the cell, or making it more negative, means that you are less likely to fire an action potential. So that's where the excitatory or inhibitory part comes in. Excitatory means I'm depolarizing the cell, and I'm moving it closer and closer to that threshold of excitation, getting closer and closer to the trigger point of starting a new action potential. Because after all, that's the only way I'm going to get the second neuron, that post neuron, the neuron on the postsynaptic side, that's the only way I'm going to get it to send on my signal. What good is it to send a signal to it if the next neuron's not going to do anything with it? So if that signal is important that I'm sending, and it needs to be transmitted on throughout the body, then I need to make that second neuron also have an action potential so it can also send that information on. So I'm going to excite it. Doing that, I'm going to depolarize the cell. I'm going to make it more positive, bringing it closer and closer to that threshold of excitation so that that second neuron will have an action potential and send the signal on. On the other hand, maybe what I really want to do is I want to tell that second neuron to shut up. I don't want you to talk. I don't want you to send the pain signal on. I don't want you to inhibit that motor neuron. So you can actually inhibit an inhibitor. We'll get into those later. So then I want to inhibit the neuron. And by inhibiting, what I'm going to do is I'm going to make it more and more negative. Because the more negative I make it, I am throwing it further and further away from that threshold of excitation, making it less and less likely that it's going to be able to fire an action potential. And that's the basic way that we communicate, that our neurons communicate. They're either going to excite something, making it more likely for that second neuron to have an action potential and carry the information on, or I'm going to inhibit something and make it less likely for that second neuron to have an action potential so that it does not send information on. Stop and go. That's the two basic signals that we have. So, whether or not in any given synapse we're going to have an excitatory or inhibitory signal depends on what type of ion channel we're dealing with. Just knowing that dopamine is being released at a synapse doesn't really tell me anything about whether that's excitatory or inhibitory. Even something like glutamate and GABA, for those who maybe have had this before, who already know that they are 99.99 always either excitatory or inhibitory, it's not still always 100%. But what is 100% is if you know what the ion channel is. If the postsynaptic ion channel is sodium, that means positive sodium is coming in and you're going to have an excitatory postsynaptic potential. If the postsynaptic ion channel is potassium, you know potassium is going to leave the cell and it's going to make it more negative. And if that postsynaptic channel is chloride, more chloride is going to come in. It's going to make the postsynapse more negative. So potassium and chloride will inhibit the postsynapse, while sodium will excite the postsynapse. So that's the most important thing. If you want to know if this is going to be an excitatory or an inhibitory postsynaptic potential, the most important thing to know is what the ion channel is. And that'll tell you everything you need to know. Metatropic, ionotropic, it can go either way. Any given neurotransmitter can go either way. But the ion channel, that's the most important key. All right, so we get these postsynaptic potentials. We're either excitatory or we're inhibitory. Now, ultimately, they need to communicate with what area of the neuron? Where did all this start? When we started talking about the action potential, where did all that start? No. Yeah, what part of the axon? The axon hillock, right. The base, that base there of where the axon and it meets with the cell body, that's where we have that high concentration of voltage-gated sodium channels. And so ultimately, that's what needs to be communicated to. Because if we're going to excite that next neuron, then we have to trigger those voltage-gated sodium channels there at the axon hillock to start the whole process so that that second neuron makes an action potential and sends it on. So basically, all of these postsynaptic potentials are just added together. They're going to be little excitation, little bit more, boom, hit. Or they're going to inhibit and take it further and further away, and once it stops being inhibited, it'll go back to its resting membrane potential. Or it'll get some excitatory and some inhibitory, so it'll add a little, take a little away, and it'll just go until it goes one direction or another. So whatever the predominance of EPSPs at the axon hillock will result in the action potential. But if those postsynaptic potentials do not reach this threshold of excitation, if it doesn't reach this point, so let's say we're depolarizing the neuron, but then it stops being excited, then it's just simply going to go back to its resting membrane potential. So it can be excited a little bit more, medium bit, a lot bit, until the point where it hits the action potential, or anywhere in between. But if it doesn't reach that threshold of excitation, then it's just going to simply return back to the resting membrane potential. The same thing for the inhibitory, that you can inhibit a little bit more, or what have you. Of course, it's never going to hit any kind of threshold there, so it's never going to be able to do anything. It's just, you're just preventing it. You're just bringing it further and further away from that threshold of excitation, making sure that this post-synapse here never communicates. This postsynaptic neuron never sends a signal. Now, there's two ways that it can communicate, and I have a little video clip that kind of better shows that than I can really explain, if it will open up. Nope, I already showed you that one.
An action potential, red dots, propagates along the axon of neuron 2, leading to an excitatory postsynaptic potential in the target cell. The depolarization spreads passively within the target cell until it reaches the axon hillock. Since the depolarization is lower than the excitation threshold, an action potential is not generated, and resting potential is resumed. Successive activation of neuron 2 at short intervals brings to summation of the excitation in the target cell, a process known as temporal summation. If at a given moment the depolarization at the axon hillock exceeds the excitation threshold, an action potential is generated. Action potentials arrive from neurons 1 and 2 at long intervals. The excitation induced by neuron 1 diminishes before the excitation induced by neuron 2 arrives. No action potential is generated. When both neurons simultaneously excite the target cell, the excitation builds up, a process known as spatial summation. If at a given moment the depolarization at the axon hillock exceeds the excitation threshold, an action potential is generated. An action potential propagates along the axon of neuron 3, leading to an inhibitory postsynaptic potential in the target cell. The negative charge, blue dots, spreads passively within the target cell, causing the cell to hyperpolarize. The membrane potential declines further from the excitation threshold. Action potentials simultaneously arrive from an excitatory neuron 1 and an inhibitory neuron 3. the excitatory potential merges with the inhibitory potential, so that the membrane potential remains unchanged. An action potential arrives from neuron 3 and causes the target cell to hyperpolarize. When the membrane potential begins to return to its resting potential, excitation is simultaneously triggered by neurons 1 and 2. The temporal and spatial summation of these potentials initiate an action potential at the axon hillock of the target cell. The terminal bouton of neuron 4 synapses on the terminal bouton of neuron 3. This type of synapse is termed axoaxonic. The action potential arriving at terminal bouton 4 leads to an inhibitory postsynaptic potential in axon 3. An action potential moving down axon 3 reaches the hyperpolarized area and dissipates. This prevents the release of neurotransmitter from the terminal bouton of neuron 3 to the target cell.
So you can even have this synapsing happen not necessarily on the dendrites. You can actually have synapse onto the axon of another neuron, so that you can, in this case, you'd be inhibiting the inhibitor. You would say, okay, I know that this is an inhibitory neuron that's going to send an IPSP to this neuron, but I want to turn that off. So then another inhibitory neuron can actually then prevent this neuron 3 here from even releasing its neurotransmitter, so that the target neuron never even receives that. But the two main things, we have temporal summation and spatial summation. Temporal summation means that one neuron is hitting the next neuron quickly, so that the signal, the EPSP, starts to build up. Because when that signal travels across here, the cell body is big, and it's going to dissipate. All those sodium ions are just going to go their different way. But we need them to build up and have a depolarization here at the base of the axon. So we have to somehow make sure that that signal is actually getting all the way from one side of the neuron to the other. So one way to do that is for one excitatory neuron to hit that target neuron over and over quickly. And so instead of having a slow occasionally and let EPSP die back down, EPSP die back down, if you start doing it quick, then they'll start building up. They don't have time to die off, and they'll start adding up to hit that threshold of excitation. The other way to do that is through spatial summation, where it's not necessarily one neuron hitting the target neuron fast, but you have multiple neurons all talking at that target neuron all at the same time. So that you're going to have that same effect. Each one's going to give a little bit, but then it'll all add up to excite the neuron. Okay, let's see. We've already talked about this part here. So if we're dealing with a sodium ion channel, then sodium will come into this cell and will depolarize it. If we are dealing with a potassium ion channel, potassium is going to leave and is going to then hyperpolarize the cell for an IPSP. If it's a chloride channel, chloride will come in, also hyperpolarize the cell. And if we're dealing with calcium, well, calcium, that's really, when anytime we're dealing with that, it's gonna have some other kind of physiological effect. It's not really plays a role in EPSPs or IPSPs, but it's going to usually activate some type of enzyme or something to cause other cellular processes to happen, like the terminal boutons leaving. So if you remember back last week, I said there's two types of signals we're going to be dealing with: analog signals and binary signals. The action potential is an example of a binary signal. There is no difference really between one action potential and another. An action potential is an action potential. It's going to go down to the end of the terminal bouton, and it's going to trigger voltage-gated calcium channels. That's what it does. But there's nothing from one action potential to another. There aren't little action potentials and big action potentials and middle. It's just an action potential. On the other hand, if we're looking at those synaptic potentials, the EPSPs and the IPSPs, those are gradients. Those you can have a little one or a middle one or a big one, with either the EPSPs or the IPSPs. So that is a analog signal, because there isn't just one EPSP or one kind of EPSP or one kind of IPSP. So synaptic potential is an analog signal. The action potential is a binary signal. Synaptic potentials are due to chemical messengers. We're going to open up chemically gated channels. The neurotransmitters are going to bind and open up those channels. Whereas the action potential is an electrical signal. Because here we have the depolarization from the EPSPs, which are going to trigger the voltage gated ion channels. The synaptic potentials are passive. There's no cellular mechanism that actively sends the signal from the dendrites to the axon hillock. It's just passively being propagated. Yes, ma'am. It's all a circle. So you need synaptic potentials to create the action potential, which is going to travel to the end and going to cause release of neurotransmitter, which will create the opening of new synaptic potentials. So this is that circle where I said we got to start in the middle somewhere. So synaptic potential, and this part of the action potential where I drew the little line going up to the threshold of excitation, that's EPSPs going on right there. And then the action potential happens, and then that's going to trigger the release of neurotransmitters, and if there's enough of that, then that's going to trigger synaptic potentials on the next neuron, which may or may not then create a new action potential, which will keep going on. Does that make sense? Okay. So synaptic potentials are passive. That's why you need either the spatial or the temporal summation in order to get enough of that signal from the dendrites to the axon. Whereas the action potentials are actively propagated. There's cellular mechanisms in there. Whether you have unmyelinated and it's just constantly being regenerated every step of the way down the axon, or whether it's a myelinated neuron and you have those nodes of Ranvier that are going to act as signal boosters, there's still an active measure to propagate the action potential down the axon. This is just an example of this. This was a friend of mine working in a lab with me in Boston, and I said, hey, can I have an example of a real action potential so I can show a class? And this is the figure that she gave me, the data that she collected. And this was a neuron at its resting membrane potential. Then she hyperpolarized it, and then she excited it, though this isn't typical because this is an experimental situation. Basically, she zapped a neuron and made it do this, and so she just turned on the voltage, and that's the reason there's this kind of weirdness to it, how it looks. But this also shows you how quick we're talking about. So this is 500 milliseconds. And we've already had time for 1, 2, 3, 4, almost 5 action potentials to go off. So, this is a very small time frame that we're talking about here. So how does this all end? I was saying it's a cycle. You have one thing, so something has to stop it all. So how does the signal end? How do we terminate synaptic transmission? One way that this can happen is through ion channel inactivation. Basically, the postsynapse, the ion channels on the postsynapse can get worn out, where they just simply can't be activated again. So it doesn't matter what neurotransmitters keep being released from the presynapse, they're just not having it anymore. They just can't do their job anymore, and so they can get worn out, and so therefore, even though neurotransmitters are coming through, they're not sending EPSPs or IPSPs onto the postsynapse. So that's one thing that can happen. Another way is through the enzymatic decay of the neurotransmitter. So when the action potential reaches the end of the terminal bouton, it's going to trigger the release of a lot of neurotransmitter. So something needs to happen to that neurotransmitter, because if not, it's just going to hang out there forever. And so one thing that can happen is that there are enzymes out there that will eat, basically, those neurotransmitters and will deactivate them. So then they can't keep signaling the postsynapse. That the enzymes can destroy the neurotransmitters so that they can no longer signal the postsynapse. Another way neurotransmitters can disappear is through the reuptake of neurotransmitters. Basically, your neurons often recycle the neurotransmitter. So after it releases the neurotransmitters, many of them have mechanisms that can grab that neurotransmitter and bring it back into the presynapse, repackage it into a new synaptic vesicle, and reuse it. So that is the reuptake. The most popular class of antidepressants is serotonin reuptake inhibitors. That's what it does. It prevents the reuptake of serotonin back into the presynapse. Another thing is what we call just the uptake of neurotransmitter. So the reuptake is the neurotransmitter going back into the presynapse. The uptake of a neurotransmitter would be a different cell taking it up. So glial cells can scavenge and can then take in neurotransmitters. We can also have the internalization of a receptor. In other words, I talked about this earlier, if we are getting signaled too much, your body might say, okay, we need to remove receptors out. So it can pinch off and grab receptors and bring them inside so that they can't be signaled anymore. And then we can also have what we call autoreceptors. Autoreceptors are receptors, so we've only talked about postsynaptic receptors, but we can also have neurotransmitter receptors back on the presynapse. And the basic function of this is it acts as a regulator. Because what it's saying is that, okay, if there's so much neurotransmitter flooding into the synaptic cleft that it's starting to come back and float over here, so that it comes back and starts talking to the presynapse, then hey, maybe we've released too much neurotransmitter. The barrel is overflowing with water. And so this serves as a signal to say, okay, cut it off. We've flooded the system. We don't need any more neurotransmitter. Yes, the autoreceptors would trigger the presynapse to stop releasing neurotransmitter, or at least decrease the releasing of the neurotransmitter. And then I guess there's just plain diffusion. I mean, your body's big. Neurotransmitters are small. So some of it's just going to float away. So that's also what happens. So this is just kind of a summary, once again with the binary and analog, showing how we have an analog signal coming into the neuron until we form the action potential. The action potential is a binary signal, which then triggers a gradient response. Remember, not only are the EPSPs and IPSPs gradient responses, but the amount of neurotransmitter being released is a gradient response. Because it can release a few molecules or a bunch of molecules or flood the system. So that has an all or nothing, a gradient response also. So the postsynaptic potentials and the neurotransmitter release are analog signals, where the action potential is the binary signal. Now here's a question to you. Let's say that I want to transmit something that is an analog signal. Let's say that I want to transmit temperature. So I can touch a plate, and that plate can be cold, or I can warm it up a little bit, or I can get it to be room temperature, or I can make it a little warm to the touch, or I can make it very hot to the touch, at any temperature in between. So I have these action potentials. And I said there's no difference. One action potential looks just like another action potential. There's not a cold action potential. There's not a hot action potential. So how can I take a binary signal like an action potential and convey analog information, like what temperature something feels like? How could I do that? That's the quickest anyone's ever answered that question. The frequency of it. Because remember, the action potential is what's going to trigger the release of neurotransmitter. So imagine if I've got a big barrel that has a hole in the bottom of it. And the reason I have a hole there is because neurotransmitters leave, the enzymes decay, they get reuptake, they don't hang around the synaptic cleft forever. So imagine the barrel is the synaptic cleft, and I'm going to have buckets, and I'm going to have buckets with water in, and the water's going to be the neurotransmitter. So if I go and I take one bucket and I dump it in there, and I come back over here, water's flowing out the bottom of the bucket of the barrel, I'm going to fill up another one here, I'm going to put some more neurotransmitter in there. There's never going to be a lot of water standing in that barrel, is there? Because it's going to keep flowing out, and I'm going to come and I'm going to put a new bucket in there. So there's never going to be a large amount. Now, if I want that water level to start building up a little bit in that barrel, then I come and I start doing it a little faster. Take the water, pour it away. Now, It's still flowing out. It's not going to really build up high, but some of it's going to still be in there because I'm going to be putting it in faster than it's flowing out. So I'm going to start building up a little bit of level in there. And if I want to build a lot of level in there, then I'm going to start doing it really fast. And I'm going to be able to build up that water level even more. So how fast I start dumping water into that barrel will tell me how high I can build up the level of water in that barrel. And so it's the same thing with the action potential. If I want to communicate, if I want just a little bit of neurotransmitter to come out, I can send slow signals. And if I want, and then I can start doing it faster, and there'll be a little more, and I can start. So I can control the analog signal, bless you, by the frequency of the binary signal. Does that make sense? Can you follow that logic? That the faster the action potentials come, the more neurotransmitter I can dump out, which means the more EPSPs I can create, or the greater depolarization I can cause, which will then send more and more action potentials on the second neuron. So if I want to indicate intensity, then I need to send a high frequency of action potentials. And if I want to indicate something on the low side of the analog, then I would send a slow frequency. Or, because that's what I can modulate, because all the action potentials are the same. All action potentials are the same. Whether I'm talking about information coming from my eye, or coming from my fingertip, or coming from my ear, or my nose, they all look the same. An action potential is an action potential. But I can modulate that response. I can modulate the analog aspect of it by modulating the frequency of the binary signal. All right, so let's go ahead and do this. Let's go ahead and take a short break, and then we can. So I glossed over a whole big part of this, which was the neurotransmitters. And so now for the next several weeks, we're going to now talk about neurotransmitters. So let's take a few minute break, and then let's start talking about some neurotransmitters.